Okay, so here we have a eukaryotic cell. I know this is a eukaryotic cell immediately because I see this big nucleus and all of these organelles here. And let's start off, let's look at this nucleus. The nucleus is where we have DNA. It's the DNA is stringy like this. But if we were to take a closer look at this DNA, if we were to really zoom into that DNA there, what it actually looks like is it's a double helix. Like this. But we'll talk more about DNA later. For now, let's continue on with what this stuff actually is. So first of all of this space around here is the nucleus. We can kind of zoom out and see a little bit better. All of this is the nucleus where we get the DNA we inherit from our parents. And then next we have nuclear envelopes. So that is all of this purple stuff here. That is essentially it's a cell membrane very similar to the cell membrane found here on the edge of the cell, but it's found here in the nucleus. And if you don't remember what that is, we can talk about lipid bilayers again. So a lipid bilayer is just, let me get a different color here. It's just two layers of lipids. And so one lipid sticks in and another layer sticks in, and then the layer just sort of goes like this. And if you remember, part of that, the inside is hydrophobic. It repels water, and the outside is hydrophilic. It can attach and adhere to water, and this helps create a water barrier and separate the nucleus from the rest of the cell. So that's the nuclear envelope, and then we have nuclear pores. If there is a barrier, there must also be passageways. And so what comes through the passageway here is a copy of this DNA here. Essentially, when it's time to read DNA, you'll read a small little portion of that DNA, and it then makes its way out of the nucleus, where it is used as information to perform certain tasks for the cell. So essentially, this is how information in the nucleus here gets out to the rest of the cell. Then we have one more thing here, and this is called the nucleolus. The nucleolus is essentially just DNA, but it's like very tightly wadded. It's like a big wad of DNA, and it's all concentrated information used to create ribosomes, which are extremely important for cells. You can see here's a ribosome here. They make their way out here once they've been read inside of the nucleus, and they sort of rest around the nucleus because their job is to actually read information and create proteins. They are the builders of the cell, so we have a lot of DNA dedicated to them. Okay, we're going outside of the nucleus, and the first thing we're going to see is the rough endoplasmic reticulum, again, where our ribosomes like to hang out, as well as on the nucleus, the nuclear envelope. The ribosomes want to be as close to the nucleus because there's RNA coming out of here. The RNA is coming out of the nuclear pores, and the ribosomes are exactly what are going to read that information, and they're going to turn it into protein. So they're going to be right on there, and the rough endoplasmic reticulum is where they want to work. So if the ribosomes are like workers of the cell, the ER here, the rough ER, is where they like to do their work. Now there's places on the ER that there are no real ribosomes. Let me get rid of this guy here. And that's the smooth endoplasmic reticulum, where there are no ribosomes. The basic job of the smooth is to just make lipids for like lipid bilayers and stuff like that. Okay, now we're going to move on to another organelle, and this is the liquid storing organelle, the liquid storing organelle, and that's the vacuole. And I have a picture here to represent two different vacuoles. So first of all, you have an animal cell, which is what we're dealing with in our picture. And you can see the vacuole here is very small and numerous. There's a few of them. Whereas if you go over here to the plant cell, the vacuole is very large. And that is because plants need a lot more water. And the reason for that is this vacuole, when it's filled with water, will secrete a pressure called turgor pressure. This turgor pressure is going to inflate the cell. When you don't have enough water, the cell becomes wilted, because there's just no water keeping it inflated. So again, for plants, we call it a central vacuole. It's one of the fastest ways to tell that you have a plant cell. Okay, on to the next. We've got the Golgi apparatus, also called the Golgi body. It's a folder and a shipper. And so what I mean by folder, when I say folder, I don't mean like a folder you put your papers in. I mean like a clown who folds balloon animals. So essentially their job is they receive, once these ribosomes here have made polypeptides, a string of polypeptides, the Golgi apparatus is then going to fold it and maybe even like add some extra stuff on. And that creates a protein. So they finish making proteins, and then they're also going to send them to their final location, in these vesicles here. Vesicles are shipping containers. If this is a shipping facility, these are the shipping containers that they send things out to. And usually the way it works is they have like a little address, a little receptor of some sort, like maybe a carbohydrate. That's like a, let me use a different color. That's like a lock and key. So it's looking for an opposing receptor like that to attach to. So maybe this vesicle here has a protein on it that's meant for this organelle here called the mitochondria. And the way that would work is a receptor on the mitochondria would fit the receptor on the vesicle after it came from the Golgi. This is how things get around in a cell, through receptor-mediated attachment. And the vesicles are a very big part of that. Nothing is magical in cells. It's all very mechanical. Okay, so next we have the mitochondria, which is often called the powerhouse of the cell, but I prefer to think of it as more of the battery charger. So you know how there's those green batteries that you get? They're a little bit different than like regular batteries, and that with a green battery you can drain it and completely use it up, but then if you plug it in overnight you can like start to refill it, right? And then you get to reuse those batteries. Well, cells also have batteries, and we call those batteries ATP. Now we have a whole chapter on ATP, so I'm not going to get too much into it right now. That's in the next chapter. But ATP is like a cell battery, and these bad boys here, these mitochondria, they actually assist with that. Now what's really interesting about mitochondria is they actually have their own DNA. Now not a lot of people know this, not even a lot of science teachers know this, but it turns out that when you look at mitochondria, not only do they have their own DNA, but it's circular DNA, right? So circular DNA looks like this. It's, you can imagine it squiggling around, and then of course it's double helixed, right? So they have circular DNA, and this circular DNA is exactly the kind of DNA that we find in bacteria, which has raised a very interesting theory, or I guess I should say hypothesis, and that is the hypothesis of endosymbiosis. So it is in fact believed that the mitochondria here, the way that they were incorporated into our cells is they were actually eaten by our ancient ancestor cells. And instead of us digesting them, we gave them a home. They make energy for us, or rather they charge energy for us. And in exchange, we give them a nice, safe place to live and a constant supply of food. We have a symbiosis with them, a relationship. Other evidence of this is that there's two membranes, because we ate them and we enclose them in our own membrane, but they also have their very own membrane as well. So that's more evidence. And other evidence is that they actually divide. They divide independently of us. So if they are ready to, they will start budding off of each other through a process called binary fission, which is also similar to how bacteria operate. So this is all evidence that they were at one point perhaps bacteria, and today they live inside of our cells, multiplying and multiplying to fill our energy demands. And what's also interesting is that if you are an athlete, you will make, not only will you make more mitochondria, but they will also be more powerful. Okay, next is one of the most interesting organelles here, and that is the centriole, a very mysterious organelle that researchers are still puzzling together. Essentially what this guy does is it sends out little tubes, and we call those tubes, by the way, microtubules. These microtubules stretch all over the cell, and what they do is they kind of hold the cell together. But in addition to holding the cell together, they also ship vesicles from the Golgi apparatus. You see, I told you nothing is magic in a cell. Instead, everything is very mechanical and very physical. And so what's happening on these microtubules is there is a special protein that is attaching to them called kinesin, which I'll draw in, I don't know, green. Kinesin attaches, and it actually walks on these microtubules. And when I say walk, I mean it literally walks on these microtubules. I'll try to attach a video for you to check out. It is pretty cool. You literally have walking proteins walking around the roads of your cells, carrying shipping containers. Everything in the cell is physical. There's no magic. What might be magic is these guys. They're often called the soul of the cell because they're really mysterious in how they operate, and scientists are still trying to figure out how exactly this works. The magical centriole. Okay, next we have the lysosome. The lysosome is one of my favorite organelles because I think fasting is completely underrated. Oops, I'm getting ahead of myself here. Let's go back. The lysosome is filled with, guess what? Lysozyme. So hopefully you can guess what zyme means. It means enzyme. Now sometimes enzymes end with ASE. In this case it just straight up ends with zyme, so it's a little bit easier to understand. But lysozyme is an enzyme which breaks down dead materials. So when something like the mitochondria might be starting to get damaged, this guy will recycle it and make sure that we're not causing any damage, and we are we're going to do that through autophagy. Autophagy is the breakdown of dead cellular material to recycle. It's really important that you every now and then give your body the chance to go through autophagy and access these wonderful organelles. Otherwise, your cell components will wear down and you will age more rapidly. And that is why almost every religion, almost every philosophy in the world, they all have some sort of fasting incorporated into them because it is just so healthy to give your lysosomes a chance to do their job. Okay, next we have the flagella, and here you can see I have a little GIF here of a flagella. They work through a rotary machine powered by ATP batteries from your mitochondria, and they just sort of like flap around. And what they do is they're going to propel the cell. Now, only one cell in the human body actually has a flagella, and that is sperm. It's a tail-like structure that propels the cell. Okay, now let's look at something smaller, and just to give you an idea of scale here, I'm going to zoom out again. So this is your cell, and if you remember when we looked under the microscope, we saw the difference in size was like this would be bacteria. So bacteria, which are called prokaryotes, they are so much smaller than eukaryotes. They cannot fit things like a nucleus inside of them. So just remember that. So if we look at these guys, oh, hold on. I got to zoom back through here. This is going to be a little frustrating. I'm sorry. I'm not just going to re-record everything because of one little mistake. So let's keep going. Let's keep going. All right, here we go. Here's our bacteria friend. You can see he's much smaller. I really wish that would have scaled. But you can see the size difference here. Much, much smaller. Okay. All right. Let's just trying to show scale. Okay, so this is our bacteria friend here, and you can see he's too small to have a nucleus. So he's not going to have a nucleus at all. He is what we call a prokaryote. So what you saw before was a eukaryote. Eu and karyote. I'm running out of room. So eukaryote, EU means true, and then prokaryote, pro means before. So this is a prokaryote. meaning it existed before a nucleus existed. Right? So this does not have a nucleus, and it also has extra stuff like it has cilia. Cilia are hair-like structures. We have some ciliated cells, like for example in your esophagus, which help to move mucus. But other than that, we really don't have any ciliated cells. That's mostly for single-celled organism. This is one cell. It's only a single cell. You can see it also has a flagella in this instance. So what's smaller than a bacteria is a virus. Viruses are so small that they don't even have ribosomes. Now, if you look back for a second, you can see that the bacteria is small, but it's at least big enough to have ribosomes, which means it can make its own proteins. So this thing can reproduce itself. Whereas this guy here, it can't make more of itself because it has no ribosomes. Remember, ribosomes are the builders of cells. This guy doesn't have them, so he's not technically classified as alive. So the virus is not alive. This particular virus, it's actually my favorite virus. Viruses don't always look like this. Sometimes they look like COVID looked like this, and it had a little shell around it. But this guy is my favorite virus. He's called a bacteriophage. And the reason I like bacteriophages is because they don't affect us. They affect bacteria. And in fact, if it weren't for viruses, we would be quickly overrun. They kill about 40% of them in the oceans every day. So they're extremely important for keeping bacteria levels down. That's our virus. Okay, I hope that gave you an idea of scale. And that's our overview. That's pretty much cells in a nutshell. Later we're going to get into we're going to start getting into more details. So we're going to start looking at things like the mitochondria and the cell membrane next. Thanks for tuning in.